Hello everyone, my name is Mark Maresca, and today I'm going to give the lecture about the use of Mediterranean food waste to find new phytochemicals with health effect. This lecture was jointly elaborated with the University of Novi Sad in Serbia. Um, this lecture is part of the lecture from AGRIMA Erasmus Plus program, AGRIMA for Agri-food Waste Management for Sustainable Bioeconomy through Higher Education Curricula and Upscaling. AGRIMA is funded by European Union and aims to foster university capacity to build a green transition through innovative practice and higher education curricula updating in agri-food waste management for the circular bioeconomy. AGRIMA program involves different partners. We have AVIP, University of Porto, as well as University of Novi Sad, University of Agen, Web2Learn, as well as Aix-Marseille University. My presentation today will be about the use of the Mediterranean food industry byproduct and waste as a source of phytochemical with health effect. What we call Mediterranean diet or Mediterranean food industry diet is the use of particular diet known to improve the health of people. When you look at the detail of the Mediterranean diet, you will see that most of the intake is based on vegetables with low intake of meat and processed food. And the Mediterranean diet is known to be good for you and for Earth, as it costs less energy and have less environmental impact during the production. It's also good for your wallet because the vegetables cost less than the meat. What we know from the Mediterranean diet is that it's protecting the body of the consumer. So when you eat the Mediterranean diet, you have an improved microbial diversity and as well as a protection against different diseases, mainly linked to metabolic disorder, diabetes, obesity, and also cardiovascular disease and cancer. And when you look at the number of deaths per year, you will see that the Mediterranean diet is able to prevent quite a lot of the important disease causing a lot of deaths per year. And how it works? It works because the Mediterranean diet contains a lot of phytochemicals able to improve your health and protect you from disease. For example, we know that the Mediterranean diet is able to limit or to prevent the development of obesity and of the development of diabetes. Both of them are able to cause lately cancer in humans, meaning that when you prevent obesity and diabetes, you have a way to prevent also cancer development. And when you look at what is inside the Mediterranean diet compared to other diets, you will see that the Mediterranean diet contains a lot of vegetables and fruits that are very good as anti-cancer molecules, or diet at least. And same contain also olive oil that is very good to protect you from the metabolic disease, obesity, diabetes, and cancer. Contains also whole grains and dairy products that are also able to protect you. The good point with the Mediterranean diet is also that it's low poor in fish and meat that are both bad for your health. The situation with the red wine is more complex because the data from the literature are not very clear about the protective or detrimental effect of the red wine in the diet. Looking at what are the molecules involved and how they work, the Mediterranean diet contains a lot of polyphenol, phenolic compounds, fiber, and phytosterols, as well as polyunsaturated fatty acid, all of them being able to protect your body from injury, from the diabetes, obesity, and cancer. And when you look at the mechanism involved, it's quite well known that all these molecules, the phenolic compounds, the antioxidant, the unsaturated fatty acid, are able to play a role, or at least to modulate important signal pathway involved in the cancer prevention. Okay, so the fact that the Mediterranean diet is able to protect the people from disease is well known now and is admitted. What about using the Mediterranean food industry byproduct and waste as a source to generate and to extract interesting molecules for health application? As you know, millions of tons of waste are produced by the agriculture every year worldwide, including by the Mediterranean food industry. And this byproduct and waste consists of non-edible part of the food, such as peels, steam, seeds, pulp, among others. And this non-edible part may have similar content of active molecule as the edible part that are consumed. And the question is, are these byproduct and waste that are cheap and easy accessible in large amount can be used as a potential source of molecule with health application in pharmaceuticals, nutraceuticals, or even in cosmetics? And to illustrate this point and to address this question, I take some example of byproduct and waste from the Mediterranean food industry, taking as an example olive and olive oil, tomato, tuna, and pomegranate. Starting with the olive oil and olive, the byproduct and waste from olive and olive oil are in tons every year. Just an example, when you look at the production of what we call olive oil meal wastewater, that is resulting from the process of washing and crushing the olive to produce the oil, it's estimated to be around 20 million tons per year everywhere in the world. And the question is, can we use this oil, olive oil meal wastewater as a source of active molecule? And clearly the answer is yes. When you look at the amount of interesting and active molecule in olive oil and in residue waste byproduct of olive oil production, you can see here that, in fact, a lot of active molecule are present in the waste, a lot. And when you look more in detail what are the type of waste containing this active molecule, you can see here that the olive oil meal wastewater is an important one, but it's not the best one, to be honest. Pulp and seeds contain even more of this active molecule. And what we know from these active molecules that are unsaturated fatty acid, tocopherols, and other phenolic compounds, it's already known that they possess quite a lot of active activity, biological activity, such as antioxidant, anti-inflammatory, anti-diabetes, playing a role to protect against hypertension and to protect against cancer. Here I take some example about cancer. And you can see here that it's already known, proved at least, even in vivo, using animals, that the olive oil meal water waste are able to play a protective role, even in vivo, as I say, against different type of cancer, colony cancer, lung cancer, prostate cancer, breast cancer, bladder cancer, and melanoma. And this anti-cancer activity of the waste of olive oil have been attributed to different kind of phenolic compounds that can be seen here, that are quite well characterized now, and even how they play a role as an anti-cancer molecule. what we know from this molecule is that they are able to prevent the proliferation of cancer cells and to cause their death by apoptosis. They also play a role as an anti-inflammatory molecule, and by doing this, by preventing the inflammation, they have a preventing role in cancer development. Also important and related to the two other activity, this molecule have quite strong antioxidant activity, and by this way, again, prevent inflammation and proliferation of cancer cells. What is good with this molecule also is they've been shown to be able to stop angiogenesis. That is very important during the metastasis phase of the cancer. And I will go back here. You can see that they are also been shown to be able to synergize with classical anti-cancer drugs used in medicine already, to enhance their potential of anti-cancer drugs. So the molecule by itself is active, but it's also reinforcing or acting synergistically with medical drugs used already to treat the cancer. So it's quite cool. It's quite nice. A second kind of byproduct and waste that are issued from the Mediterranean food industry is that I take as an example at least, is the one from tomato. So tomato is a lot of used in Mediterranean diet. Also is quite a lot produced in China, but it's a classical part of the Mediterranean diet. And as you can see here, the active molecule of tomato that are ferulic acid and also coumaric acid, quercetin, caffeic acid, and lycopene can be found in different parts of tomato. Not only the pulp that is already used for cooking and in the Mediterranean diet, but also the skin and the seeds that are really can be considered as waste or as byproduct of tomato. And as you can see here below, you can see that sometimes the molecules are the same, sometimes they are different, depending on the part of the tomatoes that you take, and also the amount present. So let's say, let's take lycopene as an example. You can see here that in the seed, the lycopene content is between 1.6 to 60 milligram per 100 gram of materials. When you look at the skin, it's containing much more, 10 to 100 times more, and much less in the pulp, meaning that in fact, in the case of lycopene, that is a very interesting molecule, we see it in a few minutes. Lycopene that is very strongly active as a bioactive molecule is more concentrate in the waste of tomatoes than in the part that is used in the diet itself. As I say, lycopene is a very interesting molecule, very active, and different labs have used different techniques to purify it, to extract it from the tomato, either from the tomato itself or from the waste of the tomato. And I know that other lectures already focus on this technique, so I will not take a longer time about that, but I will just enumerate them. So one technique that shows a quite nice extraction yield is the supercritical fluid extraction with CO2 that allow to use environmentally friendly techniques without organic solvent to purify lycopene. Another interesting and quite, I would say innovative, but I'm not necessarily innovative because it's not new, but it's nice to see how it works. It's enzymatic assisted extraction. When you use enzyme cascade to purify and to extract lycopene. In that case, however, you will need at some point some solvent, organic solvent or other kind of solvent to be able to extract the lycopene. So it's less environmentally friendly than the first one, than the supercritical fluid extraction. More classical because it's used from decade now. So you have the microwave assisted extraction that works with heating with a microwave to allow a better extraction of molecule from the matrix. You also have the use of mixed polarity solvent mixture that is not very eco-friendly, I would say. and ultrasonic assisted extraction, again, that is less based on solvent, but it's a more classical way to extract molecule from the matrix. Still about the lycopene. Lycopene is known to have a lot of biological activity, demonstrated, proved, not always by clinical trial, but at least even with in vivo, at least with in vivo data confirming its effect. So, for example, it's known that the lycopene and tomato extract containing lycopene are able to play a role in reducing the absorption of dietary fat, including cholesterols, but also saturated fat, meaning that tomato extract can be used to lower the plasma total cholesterol content and so by this way to avoid obesity and diabetes. It's also able to stimulate the beta oxidation, so the degradation of fatty acid inside your cells, meaning that is lowering the absorption by the intestine, and in the parallel is increasing the metabolization of fatty acid by your cell through the stimulation of beta oxidation, mitochondrial beta oxidation, so the use of fatty acid to produce energy. So this clearly demonstrates that tomato extract are able to fight against obesity and to reduce fat intake, and in parallel to increase fat used by the cells. Another interesting activity of tomato seed extract is that, for example, they are known to be able to protect against platelet activation and aggregation. Meaning by doing this effect, the tomato seed extract are able to prevent some thrombosis inside the artery and to protect against cardiovascular disease. Also related to cardiovascular disease, tomato seed extract are able to play a role as inhibitor of the ACE enzyme that is known to be involved in hypertension disease. By blocking the enzyme, you block the conversion of angiotensin 1 to angiotensin 2, that is a vasoconstrictor molecule that is causing an increase in blood pressure. Doing this, the tomato seed extract is able to prevent the activation of angiotensin and to reduce the vasoconstriction, meaning that it is able to lower the blood pressure. As a side effect, or I would say a bonus about the ACE inhibition, we know that ACE is also important for the COVID viruses. It's already known that ACE inhibitor can play a role to inhibit infection of cells by the COVID viruses. And it has been shown that the tomato seed extract, due to its effect on ACE and ECE receptor, is able to block the infection of cells by the COVID viruses. As for olive oil, tomatoes and tomato waste have been shown to be able to protect the body against cancer. And this is particularly the case for lycopene, as I say, that is very active. And it has been already shown that lycopene is able to fight different types of cancer in human, at least in vivo, in animals, and also potentially through a clinical trial showing protective roles. So this include prostate cancer, breast cancer, lung cancer, ovarian cancer, and stomach cancer at least. And all this effect of lycopene shown in vivo or even in clinical trial have been documented in terms of cellular effect to explain this kind of potential effect of lycopene. And so it is well known that is due to the anti-inflammatory and antioxidant activity of lycopene, but also on the effect of lycopene on cell division, apoptosis, and metastasis formation. Another interesting molecule from tomato and tomato waste is the quercetin. It's a well-known polyphenolic compound that is able to inhibit the inflammation, but also have anti-tumoral, anti-cancer mechanism of action based on cell signaling, because it's known that quercetin is able to prevent the activation of NF-kappa B. That is a very important translocation factor involved in the cell division. It's also able to block the expression of different enzymes involved in the inflammation, like INOS and COX-2, that are very important in the inflammation process, but also in the tumor progression. Blocking the NF-kappa B, INOS, and COX-2, quercetin have been shown to block both inflammation and cell division of cancer cells. It's also able to play modulation effect on different cell signaling pathway involved in cell apoptosis. And so quercetin is known to be able to cause apoptosis of cancer cells. It's also able to block metalloenzyme, matrix metalloproteinase to be precise, that are very important during the metastasis process because they are necessary for the cancer cell to detach and to invade other organs. So as I say, tomato and tomato waste contain a lot of active molecule, and the best one in terms of activity and about characterization are lycopene and quercetin. The third example of waste of Mediterranean food industry that can be used to obtain bioactive molecule that I decide to take as an example is tuna. So tuna is a fish from the Mediterranean Sea, but not only, it's also found in different parts of the world. And as other fish and seafood, tuna generate a lot of waste in tons. And all this waste of fish and seafood can be used to purify, to extract different kind of molecules that are quite interesting for humans. We can see, for example, unsaturated fatty acid that are very good because they have anti-diabetes, anti-inflammatory, and anti-hypertension effect, as well as cosmetic applications. From fish and from seafood, you can also extract gelatin and collagens that are quite good as antioxidant and very used in food supplement for joint pain and joint release. The seafood and fish are also a source of polysaccharide and minerals that can be used as food additive and food supplement. And my presentation today about tuna and tuna molecule extract from waste of tuna will be more focused on bioactive peptides. So tuna can be used to obtain bioactive peptides that have been shown to have different activity. We will discuss this a bit later, but just for you to know, in fact, this peptide can be obtained from almost all parts of the fish, from scales, from viscera, from head, from muscle, and from the skin. The procedure for extraction is indicated here. So fish or its waste are homogenized in water, then inactivation of internal enzyme by heat, then enzyme addition such as protease to cleave the protein and release the peptide, then followed by an enzyme inactivation, centrifugation to separate hydrolyzed protein and peptide, and then freeze drying, and then fish production of extract of fish protein hydrolysate. And when you look at this hydrolysate, they contain quite a lot of interesting peptide with strong activity. And for example, I take the example here of some peptide sequence that you obtain after digestion of the waste of tuna. And all these peptides have been shown to be able to have antioxidant activity, either by inhibition of the cell signaling inside the cell or by scavenging free radicals. And again, this peptide can be used and already used as antioxidant. Interesting also, like for tomato, some peptide that you can extract from protein hydrolysate of waste of tuna. Some of these peptides that you obtain here and that the sequence is given here are able to inhibit ACE enzyme and so to prevent all the cardiovascular disease linked to hypertension. And if you look here, you see that the IC50, so the concentration causing 50% inhibition of the enzyme, are quite wide as a range. So from 0.6, sorry, to 60 to 100 and something. So the different peptide does not have exactly the same activity, so they can be more or less active, but all of these peptides have been shown to be active and to inhibit the ACE enzyme and so to reduce hypertension. But it's not limiting to hypertension and antioxidant activity. You can see here that in fact the peptide from tuna have been shown to have a lot of other activity against obesity, because they play a role in adipogenesis, so the formation of adipocytes, the maturation of adipocytes in the body. Some peptides have been shown to have anti-inflammatory effect. Some peptides have been shown to be immunomodulatory, so it's not only to inhibit the inflammation, but they can also um, modulate the immunity of the host. Some peptides have been shown to have anti-cancer activity, hypoglycemic effect, and some of them have been shown to be able to kill bacteria or fungi, so to be antimicrobial. You have the sequence, some example at least of the sequence here that you can find in the literature. All these peptides have been shown to have this kind of activity. And so I invite you to read this review and to look at how they work exactly. Another example of waste and byproducts that can be used to obtain a bioactive molecule is a byproduct and waste from pomegranate. That is a fruit typical from the Mediterranean country. And again, different extraction procedure can be used to obtain the extract from the pomegranate. And I can tell you that one of the most interesting parts as a waste is the peel of the fruit that is really easy to obtain, is not used as food itself, it's a real waste. And from this peel, you can extract by different techniques like pressure, liquid extraction, enzyme assisted extraction, microwave assisted extraction, ultrasound assisted extraction, or soak-salt extraction. You can find from this waste, you can obtain a lot of molecule with very strong biological activity. I put here some examples of molecules that can be used and obtained and used, from the peel of the fruit. So from the peel, you can obtain dietary fiber like galactose and cellulose that can be interesting, and minerals, glycoside, alkaloids, but also other phenolic compounds that are very, very biologically active. From the peel, I put here as a record, you can also obtain vitamin that can be interesting as a food additive. The activity of waste extract from the fruit have been shown to be able to have protective effect against cardiovascular disease, cancer, infection, because some of them possess antimicrobial activity. Some of them have been shown to be able to stimulate the wood healing, so the regeneration of the skin, so with a cosmetic application. And also the extract have been shown to possess anti-inflammatory effect. Now I will present you some personal data. So they are not yet published. They will be published soon. This work was performed during a collaboration with Professor Akram Hijazi and his master's student and PhD from Lebanese University in Lebanon. And what we did during this study is to take the peel from the fruit of Lebanon and perform extraction using ultrasound versus maceration using two kinds of solvents, methanol and ethanol. or compared to water. And what you can see from here is that the yield of extraction is the lowest with water and is improving with ethanol, but is the highest with methanol extract. So the yield is depending of the solvent, and then is also depending of the technique itself, ultrasound versus maceration, at least in the case of ethanol. Then when you look at the anti-inflammatory effect of this molecule, we will go to this in the next slide, but you also need, when you want to understand how they work as anti-inflammatory, to quantify, to measure the content of phenolic compounds and flavonoid compounds. And what you can see here is that even if the yield is different, clearly, from water to methanol to ethanol, the content of the yield for the total phenolic compounds, it's almost the same, whatever the solvent is and whatever the extraction method is. This is true for the phenolic compounds, but it's not the case for the flavonoid one. You can see here that the flavonoid compounds are much more extracted with water solvent than with organic methanol or ethanol solvent. And this is even more surprising when you compare with the total yield of extraction. So meaning that with water, you have like 20% yield extraction, but you have like much more phenolic, flavonoid content compared to the solvent. Next, we look at the antioxidant effect of these different extracts using the DPPH assay. As you can see, the different kind of extract, depending of maceration against ultrasound-assisted extraction, give almost the same profile. So the technique of extraction doesn't play a clear role on the activity, the antioxidant activity. Now, looking at the different type of solvent used, you can see here that the water and the ethanol solvent give the best extraction in terms of IC50 on the DPPH assay, meaning that the ethanol, ic, and water extract are more antioxidant than the methanol one. We next look at the anti-proliferative, so anti-cancer effect of these different extracts that we obtain. And for that, we focus first on, I will not show you all the data that we have, but we test different cell type. And the gastric cancer cells was one of the most sensitive. And you can see here that at different time, so each time it's here a short time, here 72 hours, here 24, 48 hours. You can see that the different kind of extract possess the ability to block the division of the human cancer, gastric cancer cells. And the effect is massive at 72 hours of incubation with a dose effect and is not correlating with the yield of extraction of the content of flavonoid, meaning that is certainly more depending of the phenolic compounds present in the extract. Another biological evaluation that we perform with this byproduct and waste from pomegranate was the use of this extract, not in anti-cancer or antioxidant, but as antiviral extract. This was based on the fact that this extract had been already shown to be able to block some kind of viruses, some type of viruses. So these tests were performed in collaboration again with Professor Akrami Jazi and his student from Lebanon, but also with Professor Shaker El Kalamouni from University of La Réunion, France. So what was done first was to look at the content and the antioxidant activity of different parts of the peel. So when you look at the peel of the fruit, you have the internal peel and the external peel. And the content of a bioactive molecule may be different between the two. So we decided to test internal and external peel with two types of solvent, ethanol or water. and using extraction. And when you can see here is that the internal and external peel extract are more antioxidant, more active as antioxidant than the juice from the fruit. We next look at the effect of the extract on the infection of human cell by Zika viruses, that is, it's a tropical viruses. And you can see here that the extract from the peel, external and internal extract, are able to block the infection of the cell at concentration lower than the concentration causing cell toxicity. It's important to compare the two. So meaning that, for example, with the internal peel extract in ethanol, you have a selectivity index of 4.5, meaning that the extract is 4.5 more active on the viruses that is toxic for the human cells. We were able to show that, in fact, the extract were not active once the cells are infected, meaning that, in fact, they possess an activity not during the replication, but against the entry of the virus in the cells. Meaning that if you add the extract on an infected cell, it's too late. The virus is able to replicate, and the extract is not able to act as an inhibitor. But when you put the extract from the start, so meaning when the virus meets for the first time the cells, you can see here that the extract are very strongly active against the virus, able to block it. Just for your knowledge, the extract from pomegranate are not only used as a health effect, they have been already used as food additive to protect the food from oxidation or other. For example, they have been used in meatballs, meat patties, in burgers, in bakery products, cake and muffin cake, to improve their antioxidant activity and to protect the food from oxidation. They have been also used in dairy products and oil production, and they can improve again the resistance of the food to oxidation during times. So in conclusion for this lecture, I hope I show you and I convince you that yes, the Mediterranean food industry byproduct and waste are clearly a source of extract and molecule with biologically interesting activities such as antioxidant, anti-inflammatory, anti-diabetes, anti-hypertension, anti-cancer, and other activity. And as mentioned previously, the extraction used during this different study and experiment have been shown that the extraction itself can play a role on the activity that you found at the end. So one important direction and remaining question is which are the best extraction method in terms of cost and benefit, environmental impact, and yield. And this will clearly depend on the waste that you want to treat, but also about the application that you want to, for which you want to use the extract at the end. Clearly, if you want to use the extract as a pharmaceuticals or nutraceuticals, we need to avoid the use of solvent, or at least to use solvents that are compatible with this kind of application. A second important issue and question remaining is what about the toxicity of this extract? Problem is that all these products from the Mediterranean food industry can contain toxins. Let's take the case of tuna and fish in general. They may be contaminated with heavy metals. Vegetable can be contaminated with pesticide and mycotoxin. And all these molecules that are toxic for human can be found in the extract. Meaning that when you extract from the waste, you can take the toxin with a good molecule and keep them until the end. Meaning that clearly we have to ask ourselves, are the extracts safe or not in terms of toxicity? The third question important to ask is, what is the best to use? Extract or pure molecule? My opinion about that, based on what I see from our own experiments, is that most of the time the extract process activity, and when you start to fractionate the extract and try to purify active molecule, you lost it. This is clearly due to the fact that most of the time the activity is due to synergistic effects of different molecules in the extract. And when you separate them to obtain pure molecule, you lose the activity because you don't have them anymore as a cocktail. So my opinion is that we should more focus on the use of the extract than trying to have pure molecules. And the last question, an important one, is again, what we want to do with the extract. Is it for animal feed? In that case, less questioning about the toxicity, or at least it's more flexible when it's about animal feed. You don't need to have such a safety compared to when it's for human consumption. So animal feed can be one application, a clear one. You can also imagine using them as human food additive, and this has been already proved to works. I'm sure that the other lecture from the AGRIMA program show you how we can use the waste from food as a food additive to protect food matrix against oxidation or microbial contamination. And we can also imagine using them as nutraceuticals, pharmaceuticals, and cosmetics. And to be honest, most of them have, most of these fields will be used with extract from waste and from Mediterranean food industry waste. I went to the shop a few minutes ago, a few hours ago, and I found this flavonoid extract from red wine, or at least from grape, used for treating legs and vascular disease of the legs. So I thank you for your attention. I hope you enjoy the lecture. And I will ask you if you can to take a minute to evaluate the lecture using the QR code here to scan it and to be able to evaluate the lecture. I thank you again and bye.